So I would like to welcome Matthew and you all to the live Q&A session of the seminar. Please ask your questions in the box on the screen, and we will ask the most popular question. So if there is one you would like to see answered, please use the voting function. Thank you so much for your talk, Matthew. So one of the most popular questions is about the organoid and how stable they are. Are they accumulating phenotypic or genomic changes when you are passaging them?
Well, that's a great question. So, well, first of all, it's a pleasure to be here. Thank you. So we have looked a lot at the organoid stability in culture and whether they acquire genetic or phenotypic changes. And we've done that in a number of ways. So in some of our earlier publications, we simply passage them over prolonged periods of time in the lab, in some cases up to 6 months. And we've taken serial samples over that duration and done things like whole genome sequencing. We've done RNA sequencing. And we can see that actually they are remarkably stable over that time. We do see some small changes, but those changes aren't things like changes in cancer driver gene mutations or large scale changes. So there's some accumulation of small genetic changes over time. We've also now looked at this from a more functional perspective, where we've grown these organoids over many months and done things like whole genome CRISPR screens in them and profiled them against hundreds of small molecule inhibitors. And in those cases, again, the evidence suggests that with respect to those phenotypes, they're very stable, that we see the same drug sensitivities, that we see the same CRISPR dependencies. What I think is important, though, is we do know that there is some selective pressure at the point of derivation. And that is probably the most important point where you see these selective pressures under the culture. But when they're in culture, they seem to be relatively stable. I will say, though, that if you are using organoids, you have to be more aware of this. And certainly you have to be very careful about how you passage and bank your organoids. And through some type of routine process, make sure that they continue to be phenotypically the same as you think they are.
Yeah, thank you so much. I guess building on that question, are you in the biobank kind of storing this number of passages and all of this? So of course it will have not a lot of influence, but still if it has some influence, you can always kind of come back.
Yeah, I mean, we have an electronic laboratory notebook and we track every parameter of these. I mean, we actually have a picture of every passage that's ever taken place and the date of that passage and all the details of all the media and so forth. So at least at the point at which we submit these into a biorepository, we have a almost perfect record of what's happening to them. And on very rare occasions, we have seen, for example, morphological changes that we are aware of and have even gone back to further investigate.
Yeah, thank you. That's very useful.
The next most popular question, Matthew, is how accurately do the organoids recapitulate in vivo scenarios? And how do you estimate the similarity between the primary tumor and the in vitro model?
Yeah, so I'll answer the second part first and then come back to the first part. So we've done a lot of work comparing the primary tissue to the organoid from which it was derived. And so, for example, we've done whole genome sequencing on matched samples. And again, we've done some RNA sequencing on the 2 matched samples. We've done histology on the samples as well. And by and large, I mean, there's quite good correspondence between, for example, mutational profiles. There's complexities, though, when you do this that do make this somewhat challenging. So, for example, when you sequence tissue, you have both tumor cells and also normal cells present within that culture, whereas whereas when you sequence an organoid, it's pure tumor. So you have a lot more sensitivity to detect, for example, rare subclonal events in an organoid that you might miss when you sequence actually a cancer sample. Equally, when you perform RNA sequencing on a tumor sample, you're getting the contribution of all the stromal components as well as the tumor cells. And again, it makes those types of comparisons more difficult. But I think our work and actually many other groups have shown that they actually recapitulate very well. And I think we're kind of very satisfied on that aspect. In terms of replicating in vivo scenarios, this is still a challenge. So we have a lot of preclinical evidence that, you see genetically, histologically, that they look more like patients' tumors. The niche factors you need to grow them reflect the actual tissue of origin better. We know that we can recapitulate biomarkers of therapy response that you would see in patients. So all that is kind of supportive. There's been some really nice data recently where they do co-clinical trials where a patient is given treatment, and in parallel, an organoid is made from that patient, and the organoid gets treated the same way. And those studies suggest, by and large, actually, the organoid shows a very similar response profile to what the patient's tumor does. So those are relatively promising. You can imagine there are some differences, though. It is obviously different growing a culture outside of the body than the response you see in a patient. What we don't know yet, and has never, I think, to my knowledge, been shown, that you can prospectively predict how a patient will respond and actually use that in the clinic. But I think those efforts will be coming in the near future. And I guess just to finally add, I think one of the beauties of the organoid system is that we can actually just finally ask this question, right? In the old cell lines that we all use, or even the PDX model that you might have in your lab, we don't have all the clinical links, and we can't compare it back often to the original tumor of origin. And so now we're really positioned to actually formally ask those questions in a robust way. And I think that alone is very exciting and an advantage of these models.
Thank you. Brilliant answer.
Yeah, I will follow up a bit on the organoids. So how many new organoids models will be needed to understand all the possible driver mutations in cancer?
I don't know. And I'm not sure anybody really does today. And it depends how you define the question. So, very naively, if you go on the World Health Organization's website and they say there's 200 types of cancer. Okay. So, ideally we would have representation of all those subtypes of cancer in a panel. But we know it's even more complicated than that. Typically breast cancer can be divided into, depending on the methodology, 3, 4, even 10 different subtypes. So you may well wish to have representation of all those subtypes. And then you think, well, I want to use these models for some form of study. And then it's probably not enough just to have one model, because how do you know that model is actually representative of that population? So you may wish to have 3 or 4 models. And then once you go beyond that, you may say, well, actually, we'd like to have early stages of disease and late stages of disease, as well as, treatment refractory disease. And so when you really start to add all this up, I think what's very clear is the number of models we have today is grossly inadequate. Now, then the question is, how many do you need? And I think I don't really know that answer, and I don't think anyone knows, but I think the numbers are much more in the tens to hundreds per cancer type, and even in cases sometimes more. That would be a more realistic number. And I think, as a comparator of that, large-scale cancer genome sequencing studies didn't sequence 10 tumors or 20 or 50. They sequenced 500. And in many cases, they're sequencing more. And they know they need to do that to get representation of different molecular subtypes. And I think the same principles apply when we start to make these models as well. We'll need hundreds, but defining that number, I think, is still unclear. But I think it's an important question that needs to be addressed in the next few years.
So I guess a bit also following on that, we are talking about variability within tumors, but what about the variability within individuals? So how many organoids do your team is deriving from the same sample you receive? Do you see changes between them, or is it, Or
Yeah.
do you think the changes are associated to the reproducibility, or is just because the heterogeneity of the initial sample?
Yeah. So one of the great advantages of the organoids is they are heterogeneous, meaning that they have multiple subclones present. And there's been some really elegant work by Hans Clevers and Mike Stratton, where they subcloned out different cancer organoids and then grew them up and showed that actually phenotypically they could be quite different, even though they were derived from the same original tumor. So, that is a whole nother layer of complexity. I think, I have a few comments on that. I think one is that as you make these biobanks, you hopefully begin to have representation of these different subtypes, whether they come from different tumors or from one tumor and different subclones of that tumor. And so you can begin to infer principles that could be applied to cover, for example, aspects of intra-tumor heterogeneity. So I think that's important. I think, additionally, when we think about treating patients and using the organoids as a model system, ideally we'll be really going after what we sometimes refer to as like truncal events. So the things that are shared by all the population within that tumor, so that we reduce the possibility of rare subclones escaping a particular therapeutic intervention. And so that's one of the ways that we can think about kind of getting around this. And I think, even beyond that, it's clear that it's going to be important in the future to begin to use some of the single cell techniques as well to understand the heterogeneity in these populations. And then when we treat them with drugs, or when we treat them with CRISPRs, for example, that we can begin to understand that heterogeneity. So I think those types of tools will help address some of these questions, but there's no question that both inter-patient, intra-tumor heterogeneity is a major challenge.
Thank you.
I've got a very specific question, Matthew, about what is the mechanism of Werner in synthetic lethality? Could you expand on that?
So this is related to the observation that through our CRISPR screens, we were able to show that microsatellite unstable cancer cell lines were dependent on Werner helicase. And actually, when we made the first observation, we didn't understand the mechanism. But there was kind of two hypotheses we had at that time. One is that Werner itself was involved somehow directly or indirectly in mismatch repair itself. And that leads to this microsatellite unstable phenotype. Or microsatellite instability leaves kind of genetic scars on the genome that accumulate over time. And in some way, Werner is involved in resolving these scars. And in fact, it appears to be this latter that's important. And this has been worked by another group that showed that in microsatellite unstable cells, you get expansions of TA repeats in the genome. Okay, and it's remarkable. This was never really realized because actually these are very difficult to sequence with conventional kind of Illumina short read technology. So you get these expansions of these TA repeats in microsatellite unstable cells. And what appears to happen is that these form secondary structures in the DNA that Werner helicase is required to resolve. And what happens is if you lose Werner helicase, these become toxic. They get converted into DNA double-strand breaks. And this induces the DNA damage and the apoptosis that you see. And so what we now have as a molecular understanding is that, in fact, microsatellite unstable cells have these long TA repeats that they require Werner to resolve, and that's why it works in these cells. And that's why, presumably, normal cells, or at least microsatellite stables, are less susceptible to Werner or less dependent on Werner.
That's great, thank you.
So, I mean, one of the questions is if the traditional cancer cell lines are now redundant for target drug discovery.
Mm-hmm. I have a much more favorable view of the cancer cell lines. I mean, they get a lot of criticism, and much of it is well-deserved, but they're enormously useful. And reflecting, I work with a lot of other colleagues who work in other disease systems and other aspects of biology where there are no model systems at all. And so these cancer cell lines, they're a wonderful gift for the community, actually. And they're very valuable. They've been used for many years. They have to be used appropriately. But I would say they have been useful because we know, for example, almost every targeted therapy that exists today his development has been supported in some way with the use of cancer cell lines. So I think for me, it's totally clear that they're useful. Now, the organoids themselves bring extra advantages. As I've mentioned, you can compare back to the original tumor. They haven't been growing in vitro systems for decades. We can track their evolution. We think the 3-dimensional culture system is probably superior. So they bring all those different advantages. But I think, we still have many applications where I think the cell lines will be useful. We think the organoids can augment that. We think you can bring these two data sets together to get bigger sample sizes that give you more statistical power. So I think, at least at the moment, I think the cell lines are still a very valuable tool. In the fullness of time, 5 or 10 years, perhaps they will become obsolete. But I think the verdict is still out on that. And I think they still have a lot of value, certainly what I can see over the next 5 to 10 years.
Yeah, and I guess a bit building on that is this 3D structure of the organoid kind of making it more complicated, all the high throughput analysis that I'm just thinking like you are growing multiple lines. And for example, if in the future you want to do single cell, then you need to dissociate all of them and they are 3D structures. I don't know, how do you see all of this?
Yeah, no, that's really a great point, Roser, and I failed to mention it. But, the organoids are more difficult to grow and they're more expensive to grow because the media constituents are more difficult to generate. And so, whereas we can routinely take a cell line and profile across thousands of molecules or very, readily, let's say, do a genome-wide CRISPR screen, the organoids are growing in this 3D matrix that's quite difficult to break apart. The cells grow more slowly. And so every step is more difficult. And so, as I pointed out in my talk, we've been working with different ways to make these screens more efficient, easier. But I still think we are some ways off making them as readily as accessible as we can with the cancer cell lines themselves.
Thank you.
Thanks, Matthew. We've got a question here is that you mentioned that known tractability, you use tractability for prioritizing the candidate genes. So does this leave a blind spot for potentially interesting candidates as drug targets?
Yeah. So, it's clear that these types of genetic screens are throwing up all sorts of new targets. And many of them are falling out of sight of the conventional target space where, existing drugs are really developed. So, for example, kinases are a major target of existing oncology drugs. And so these new target spaces are going to probably require new technologies and new ways and modalities to actually go after. And so that's part of the reason we put in this target priority score is we're trying to, find the easier ones to go after, the ones that are more practical to go after. But that said, we actually kept the tractability as a kind of separate classifier and it's not integrated into the score. For the very reason is that we didn't want to be blind to exciting targets that may just be a little bit more difficult to drug with today's technology. So if you kind of cast your mind back to the data I showed you, I showed the priority score and then we binned them by tractability because we didn't want to combine those two things. And actually, when I speak with the pharmaceutical industry, what's clear is that tractability is an evolving space. And in fact, sometimes it's a competitive advantage of certain companies over others. And so a target that one company may find tractable, another would not, and so forth. So I think, going forward, we won't incorporate it into the score, but we think it's still a valuable piece of information. But I think it is one of those things that's going to more rapidly change. And I think if we are going to want to go after these really new, exciting targets, we are going to have to think more creatively about how we actually target those new targets.
Brilliant. Thank you. Thank you.
Okay, so, well, we would like to thank Matthew again for his presentation and joining us for the Q&A session, and also for the audience who joined this virtual session. So we have confirmed that we will continue these seminars at least until summer this year. So please look out for announcements. So next month seminar is on the 17th of March, and we will be joined by Professor Matt Hars, who is the head of the Human Genetic Program. And also, well, just to remind you that these seminars and the questions and answer sessions are archived in our web pages for future viewing. And we have been delighted that this series has allowed us to share Sanger science with a diverse scientific audience around the world. Thank you so much, and looking forward to see you next month. Thank you.